In lecture, we ended up with this slide on infections that go unnoticed and those asymptomatic carriers that are kind of the scary ones. We also talked about portals of entry in the first part of the PowerPoint. Now we're going to look at, all right, well, there's different modes and different ways that things can get in the body, but what are different ways that pathogens leave the body? So various pathogens leave the body multiple ways, which then, yes, can go from portal of exit to portal of entry and infect someone else. So different portals of exit, respiratory and salivary portals, these are some of the big ones. So this is why a lot of times we collect and analyze mucus samples, sputum samples, nose drainage, saliva, all things that can harbor lots of different organisms. Epithelial cells, again, your skin and your scalp can harbor multiple various pathogens, and then anytime you touch, you pick things up, you touch something else, you drop them back down. Uh, Fecal exit, a lot of things can live in the feces, a lot of things can cause diarrhea, and they leave that way. The urogenital tract, again, various things can leave, whether reproductive, the urinary system, and then even removal of blood or bleeding. If you have a blood draw and there's a pathogens in there, well, you now have a pathogen that just left the body. And why we're concerned also, if there is blood somewhere, we have to assume that it's infectious because it could be. Depending on how long someone has some type of infection in the body, uh, we kind of group them based on what is that progression of that microbe in there. So if we say someone's in clinical latency or latency, a lot of times the initial symptoms of some type of disease have um, gone away. However, the pathogen itself is still in the body. This happens a lot with herpes, is the symptoms and signs and symptoms will come back and then they go away and then they can come back again. It's because that particular microbe stays in the body. But if the symptoms are not present, we say they're in a latent stage of that particular disease. A chronic carrier is someone who always has a latent infection, but is also shedding it. And so if someone does have, so the herpes, human herpes virus 1 that causes cold sores, well, if there are no cold sores present, we would say that particular person is in a latent stage. But if they now have cold sores and that particular virus spreads in those active cold sores, well, now they're a chronic carrier because they're shedding that infectious agent. Sequelae is when you have a long-term or permanent damage that is caused from those tissues. So I wouldn't say human herpes 1 is causing damage to tissues, but for those that may have had Lyme's disease for a long period of time, especially because it can go undiagnosed for a long period of time, sometimes there is permanent damage to tissues or organs or to nerves. So we're looking at like, well, what are the long-term effects by being infected with something? If you got sick, it had to come from somewhere. So the CDC, as well as the World Health Organization and other healthcare agencies, are tracking. Well, if where are the infections coming from? Where are they found? What numbers are they at? So we're looking frequency and distribution of diseases and other health-related factors. So a reservoir is where an organism hangs out in nature. I mean, if you got sick, it may have come from another person, but maybe it came from an animal source if it was zoonotic disease. My dog bites you, they have rabies, it spreads from some type of animal carrier to humans. Could be another human. A lot of humans are carriers for diseases. We could have picked something up by having soil contaminate your something that you ate or got into an open wound, drinking contaminated water, eating contaminated vegetation. I'm like, well, where is that particular pathogen present out in the natural world? A source infection is an individual or object specifically that we know of where your particular infection got acquired. So if I got a paper cut and it got infected, the source infection was some organism on that paper. Like we know the exact object, that where we got that microbe. If you got sick and it's because someone coughed on you, they're the source patient, they're the individual that spread that infection to you. Carriers are someone that just shelter a pathogen. They don't always show symptoms, but they can be passing that disease around. So they're inconspicuously sheltering a pathogen and spreading it. So asymptomatic carriers again are the more scary ones because a lot of people harbor a lot of different pathogens. You might not even know it that you are harboring any particular pathogens. You're not showing signs and symptoms, but you're passing it. Passive carriers are contaminated, healthcare providers that transfer various things to patients. When you're going from one patient to another, like you could be spreading a disease from one person to another on your hands, on various medical equipment. It's like you're the spreader. You're the spreader. You're doing it passively. You're not infected. You don't have the thing, but you are moving that microbe from person to person. Animals that can be reservoirs and sources of infection. So a vector is just a live animal. And yes, I know we're in the animal kingdom, but other than humans, that transmits an agent from one host to another. So a lot of them, and we'll get into them when we get into various bacteria and viruses, a lot of them are arthropods. So some types of insects, fleas, mosquitoes, flies, ticks, things that bite can spread various types of pathogens. But other larger animals can spread infections as well. Various animals can bite you and spread various diseases like rabies. Birds can have the bird flu. So there are lots of other animals. Again, anything that we pick up from a larger animal, we call it a zoonotic disease because it comes from animals. But vectors, again, you just have to think of it as something that moves something from point A to point B. Biological vectors usually participate in some type of the pathogen's life cycle. For example, mosquitoes spread malaria. They bite one infected person, they pick up that protozoan, it further develops while it's in that mosquito, and then when it bites another person, they transmit the infection. And so that mosquito actively participated in the pathogen's life cycle. A mechanical vector spreads it from point A to point B, but it is not actively part of the life cycle. An example, if a fly in the summer lands on your food at a picnic, do you know where it landed, 5 minutes prior? You don't know if that fly that just landed on your bun on your hamburger was 5 minutes prior landing on some type of feces from another animal. So if a fly lands on feces and now it picks up bacteria all over its legs and now lands on your bun, it can be transmitting a pathogen from point A to point B. But it's more of just, it's picking it up on the outside. It's not actually affected at all by, it's not affecting the life cycle of that pathogen. So again, we call zoonotic diseases if it's going generally from a larger animal to a human. A lot of times zoonotic diseases are dead-end hosts. Humans are not the final thing that these pathogens would like to be in. And part of that is that it doesn't go any farther. So, for example, if I just use rabies, because I've been using it already, if an animal bites us and it spreads rabies to us, it generally doesn't go any farther than us. There's not a lot of rabid humans going around biting other humans. Like, no, we're not thinking zombie. And I'm like, but so the rabies virus itself ends in humans. It doesn't continue on and spread from one individual to another. So we're considered dead-end hosts. There's at least 150 zoonotic diseases out there, and a lot of them are where we're finding our new emerging diseases. And it's our big concern on why we're always tracking the bird flu, as there's multiple strains found in birds. Some of them can transfer over to humans. I'm like, there's various things found in bats, because a lot of viruses in bats seem to be more easily transferable to humans. So we're looking at, what can be transferred from animal to human? Can we stop any of that? It's possible to eradicate the disease without eradicating the animal reservoir. We cannot get rid of rabies. Like, how would you get rid of rabies? We'd have to kill all the animals. That's not going to happen. You can't kill all of the carriers out in the wild. This is just looking at some of the examples that are found in animals. So rabies, all mammals could be a potential carrier. Yellow fever, various wild viral fevers, hantavirus, influenza, because yes, there's bird flus, swine flus, again, lots of different strains of the flu viruses, West Nile virus, various bacteria we can be found in dogs, ticks, birds, other domestic animals, and it continues. Anthrax can be found in various domestic animals, brucellosis, the plague, salmonellosis, tularemia, ringworm, toxoplasmosis, trypanosomiasis, trichinosis, tapeworm, scabies. I mean, these are all things that are found or potentially harbored in various animals. We pick them up in multiple different modes, whether it's spread by various insects, whether it's because we're ingesting the food itself, like salmonella, uh, but they can be spread from animal to human. We talk about diseases, whether they're communicable or non-communicable. Communicable means, it's fancy words, it can go from one host to another, meaning it's contagious. And so a communicable disease, the flu, we can spread the flu virus from one human to another. It's a contagious disease. Non-communicable means you pick it up, but not from host to host. Um, this happens sometimes when, I got a UTI, but I didn't get a UTI from someone else. A lot of times it's because, you know, my bacteria got somewhere where it wasn't supposed to be. Some of my digestive bacteria ended up in my urinary tract. It's not because I got it because someone coughed on me or I got it from someone else. So it comes from an outside source. It's not spreadable. Even rabies, I got it from an outside source. An animal bit me. Although I shouldn't say, I guess if I bit another person, it could be contagious. But let's hope that doesn't happen. Different types of contact. Direct contact is if we pick it up, if we're transmitting it. It's a physical contact, or fine aerosol droplets means you have to be in very close contact with someone. And so if they sneeze on you right in your face, we consider that a direct contact. Or if you're touching surfaces that someone else has touched, or if it's skin to skin contact, it can get spread. Indirect contact, it needs something else to transmit it. It's not direct contact. You're not in close contact with someone. So maybe you need some type of vehicle. And when you think about your vehicle that you drive, it's something that moves something from point A to point B. So maybe it's an animate material. So a desktop, some high touch surface area. Someone touched it, then you touched it, but it was the material that transmitted it. Some type of food source that came from somewhere, we ingested it, you brought some suspicious thing to a picnic, some type of water reservoir that was contaminated, a biological product that came from somewhere, or a fomite, which means an inanimate object. So anything that is usually high touch surface area, it's kind of a repeat of saying inanimate material, but usually it's high contact. Airborne, so you can get things, again, someone coughs or sneezes, it gets in the air, but things are subject to gravity. And so, yes, these little water droplets, they will fall, but there are some things when they get into the air, they do stay airborne if it's small enough. And so we usually say it's direct contact if it's, only found on the fluid part of when someone sneezes, versus if it's farther away, like 6 feet away. That 6 feet was the big COVID time, is, well, beyond 6 feet, things would be subject to gravity. It's not considered airborne at that point. So this is just showing examples of all the different ways communicable diseases could be spread. So we're looking at direct transmissions versus indirect transmissions. So fomite or contaminated objects, again, you're touching those high touch areas, and it's found on there. Fecal oral, it can get spread, it's some type of fecal matter was found on some type of food, and yes, it's more common than you think, and then you ingest it. The work of epidemiologists, and I know it's a lot of words on this one slide, again, their job is to track what is the transmission sequence. How is it leaving one body, getting into another body? And this was a really big concern and a really big job when COVID first came out because we did not know how were people getting infected. Was it multiple different ways? How was it leaving one body? What was the time frame that that was happening? And so they're constantly looking for like what happens when it leaves the body, how does it get in the body, what happens to the body once it does get in. So they're analyzing data, they're analyzing cases. They're looking at, well, are we having outbreaks? And we are right now with measles, and making sure that information gets out so people can take preventative measures to hopefully not pick up various diseases. When they look at all of this, a couple words that you may hear is prevalence. That's the total number of cases in an entire population. So if we say, well, the prevalence of, say, HIV in the United States, I want to know everyone in the United States that currently, right now, has HIV. Incidence is we're looking at the number of new cases, generally in a time period. So I want to know, well, what is the incidence of HIV? Well, how many new cases of HIV have been identified, usually in a particular year? So prevalence is like everyone that has it versus how many new cases. Mortality rate is the number of deaths due to a certain disease. Morbidity is the number of people afflicted. So, like how many people are sick. So, morbidity and mortality, similar, but one ends up with just how many people are afflicted versus how many deaths actually occur. Endemic occurrence is looking at, all right, well, where do we find a particular disease and in what particular location? And if something is always in a certain area, we say that it's endemic there. We'll talk about valley fever, but down in the southern, drier areas of the United States, there is a virus called valley fever. Not a virus, it's a fungal disease called that causes valley fever. It's endemic. It is always going to be found in these drier areas. So it's just staying at a steady pace. We're not getting like outbreaks of it. It's just always going to naturally be found there, and that's where we're going to see the cases. Sporadic occurrence means, at some point we kind of have like little mini outbreaks everywhere. We start to see cases. They're not any particular localized area anywhere, but we start to see again sporadic cases, which can sometimes happen with typhoid fever. Epidemic is when all of a sudden you're having a disease start to increase in number beyond what's expected. Like we expect some diseases, oh yeah, we normally have this many cases a year. Oh, well, now we're seeing a lot more of those cases. Maybe we're having a flu epidemic. Maybe we're having a COVID epidemic, I'm like, but we're seeing increased numbers beyond what we expect. A pandemic is when that epidemic, that increase in numbers happens across multiple continents. And that's what happened with COVID. Yes, it's happening AIDS patients as well. We're seeing increased numbers. We're seeing it global wide. But we consider that now a pandemic. This is showing examples of the COVID cases. I know it's outdated, but it's considered a pandemic because huge number of cases, huge increase in cases, and it was happening worldwide. Nosocomial infections, we're getting away from that particular word, which I'm happy because it's just a big word and no one understands what it means. And we've renamed it to healthcare associated infections. These are diseases that are acquired or developed during some type of healthcare or hospital stay. A lot of people get sick when they go to healthcare settings. It could be from the surgical procedures, so something wasn't quite sterile in that surge tech. The equipment itself, again, we talked about how tubing can be very hard to sterilize. Like, was there a biofilm on some type of equipment used? Personnel, people that are at hospitals, whether they're workers, they could be sick. They could be spreading things to people that go there. People are sick at hospitals. And so you can get diseases from other people that are at the hospital. And then the hospitals are breeding grounds for healthcare, for resistant microbes. And so this is where you can pick them up as well. So 2 to 4 million cases a year in the United States of people that are picking up diseases while in healthcare settings. There is a risk anytime you go to healthcare settings. You go there to get better, but while getting better and treating one thing, you could potentially pick up something else while there. So this is showing examples of what are different things and where are they picked up. So a lot of different things that are picked up because you breathe them in and they cause issues with the respiratory tract, or they get into the bloodstream. There was an open wound, something got in. Even when administering, putting in that IV, was it cleaned properly? Mike, or did you just put some type of microbe in there while putting that IV in? And I'm like, things can get into the urinary tract, and a lot of these are digestive bacteria. And I'm like, some things can get in and cause issues on the skin. A lot of things cause issues in the GI tract, but a big one is surgical incision and wounds. Stuff can get in. You break that first line of defense, and now you've damaged it. Things are going to get in. So in healthcare settings, we follow what are known as universal precautions. Our job is to stop infections from occurring. So we don't want patient A spreading their disease to patient B. So we don't use shared equipment. And I'm like, if you're going from patient A to patient B, you're switching gloves. You're switching anything that might be in contact from one patient to another. Um, we don't want anything from the patient to go to the worker. You don't want to get sick when you go to work. I'm like, so how can you prevent picking up anything from the patients? Again, we just assume all fluids are infectious. I'm like, we also don't want anything from the worker affecting the patient. So if you are sick, you shouldn't be going to work. Although you're going to be told otherwise. And I'm like, but you should be taking precautions, wearing masks, wearing all your proper PPE. I'm like, so we try to stop any possible infections from occurring. And we will end right there.